Hi guys. Alright, so today we're going to be talking about Chapter 16, which concerns glycogen metabolism as well as Glycogen Genesis. I decided I wanted to separate this chapter into two videos. We're still going to cover both video lectures before we meet in class next time. However, I just thought it might be a little more manageable to break the chapter up into two videos as opposed to one long video. That way you can kind of manage going over both lectures however way you want. So for this lecture video specifically, we're talking about just glycogen metabolism, which is sections 1, 2, and 3 in your textbook. So here are our learning objectives. They're a little bit shorter because we're kind of talking about one big or one aspect of metabolism, and we already got a lot of our key terms down, and we're dealing with a lot of enzymes that we've already discussed about. So let's go ahead and get started. So when we talk about glycogen metabolism, we're actually talking about a number of different processes, all of which seem to kind of coalesce into this one central metabolite, glucose 6-phosphate. So if you remember, glucose 6-phosphate is that first metabolite from glycolysis where glucose is brought into the cell and then phosphorylated to prevent it from freely exiting the cell. And so once it becomes glucose 6-phosphate, we can actually either transfer it onto glycogen if we're going to store it either in the muscle cell or in the liver as glycogen. Remember that big polysaccharide molecule that stores glucose? It can be shuttled down into the pentose phosphate pathway in order to make ribose 5-phosphate. And remember, this also generates us some NADPH. We can also undergo glycolysis to make pyruvate, which remember generates a net of 2 ATP molecules. And then pyruvate can either be used to make amino acids, it can go through the citric acid cycle as acetyl-CoA, or we can undergo fermentation to make lactate. And remember, this also regenerates our NAD+, that can then be used again in glycolysis to make more pyruvate. We can also go, we already said that, to make glycogen, or to extend glycogen. Alright, so the fate of glucose 6-phosphate really depends on the current needs of the cell. Right? So if the cell is undergoing DNA replication, maybe glucose 6-phosphate will be used through the pentose phosphate pathway, so we can make ribose, which is a precursor for nucleotide synthesis. If we need ATP, then we'll undergo glycolysis in order to then make pyruvate go through the citric acid cycle. Depending on whether or not we have oxygen present, we'll either do fermentation or oxidative phosphorylation, or if we need to synthesize some proteins, then pyruvate will be used for amino acid synthesis. However, if the cell is good with ATP, good with NADPH, and doesn't need to make amino acids, then it will be used to make, or then the glucose 6-phosphate will be added onto the end of glycogen for storage for later. So let's talk about how we get glucose from glycogen. So this is the breakdown of glycogen. Remember, is that polymeric saccharide molecule where we have branch points. Each end of the branch point we have a reducing end, and we can cleave off these glucose residues at our reducing end through glycogenolysis to produce a glucose 1,6-phosphate. So glucose is commonly stored as glycogen in granules, which are found commonly mostly in the muscle cells or liver cells. Glucose molecules can be rapidly cleaved from the non-reducing ends of glycogen using 3 enzymes. So the first enzyme, Glycogenphosphorylase, is going to cleave here or here at these non-reducing ends using a phosphate group substitution at the C1. So we'll get a phosphate group to cleave here at C1 to give us glucose 1-phosphate in a glycogen that is short 1 glucose molecule. Glycogen phosphorylase can continue cleaving until we get to about 4 or 5 residues from a branch point. And then we need a debranching enzyme, glycogen debranching enzyme, in order to move that branch and then hydrolyze at that branch point. Phosphoglucomutase can then convert glucose 1-phosphate to glucose 6-phosphate, which is that current metabolite, or that metabolite that is common among a number of glucose pathways. So let's look at glycogen phosphorylase in a little more detail. Glycogen phosphorylase is activated by AMP. So when we're under low energy yields, we have a lot of AMP increasing, then the cell wants to get more glucose, so it will activate glycogen phosphorylase to remove glucose molecules from glycogen. It's inhibited under conditions of high ATP, high G6P, or high glucose. So if we have a lot of ATP present, then obviously we don't have a demand for ATP. If there's already a lot of glucose 6-phosphate present, then why go into our glycogen stores? And same thing with glucose. If we had just eaten and we got a lot of blood glucose, and so that glucose is entering into the muscle cell, then there's no need to start to deplete our glycogen stores. To be active, this enzyme must first be phosphorylated and uses PLP, which is a vitamin B12 derivative, as a cofactor. As you can see, PLP is right here in the enzyme active site. It helps coordinate this inorganic phosphate group and acts as an acid-base catalysis to help cleave the group here at the alpha-1,4 linkage, and then covalently attach the phosphate group to that free glucose molecule. Again, as I mentioned, glycogen phosphorylase stops when it's within 5 to 4 residues. Our textbook says 4, but I've read in other textbooks it's 5 residues, so somewhere between 5 and 4 residues of a branch point. And this is because the active site is narrow and can only fit around 4 to 5 glucose molecules within the active site of the phosphorylase. So if we get too close to a branch point, it's too narrow to fit a branching point, and so it just physically can't fit and can no longer cleave at that short of a branch. Phosphorylation of glycogen phosphorylase is initiated by the hormone glucagon. So remember, glucagon is that hormone released from pancreas cells when blood sugar is very, very low. So that would indicate that if we haven't eaten in a while, or we don't have a lot of glucose in our blood, then the muscle cells aren't getting the glucose, or brain cells aren't getting the glucose. So then glucagon, or glycogen needs to start to be cleaved to release some glucose into our blood. So this happens either in the muscle or in the liver. Glycogen debranching enzyme transfers a trisaccharide, so 3 glucose molecules linked together, to an end of another branch site. So it will transfer it from the shortened branch over to a longer branch. So then the glycogen phosphorylase can then cleave these 3 saccharides because it is now further away from the branch point. The remaining 1 or 2 glucose molecules at the branch site are then hydrolyzed. Instead of using phosphate to break the group, we use water, a water molecule, to hydrolyze the alpha-1,6 branch point, or the alpha-1,4 branch point. And this uses the same enzyme. Glycogen debranching enzyme does the same thing. It does both the transferase and the hydrolysis of that glucose near the branch. And this is actually used by 2 different active sites of the same enzyme. So here we can hydrolyze that there using a water molecule. Glycogenesis is where we add glucose molecules to the end of a branch site on glycogen. So this is 4 steps One, synthesis of glucose 1-phosphate using phosphoglucomutase. So glucose 6-phosphate is mutated to glucose 1-phosphate here. So there's step 1. The synthesis of UDP glucose by UDP glucose phosphorylase. So here we take UTP and we couple this transfer reaction when UTP loses a pyrophosphate, which is then hydrolyzed to form 2 inorganic phosphates. So this usually releases around, has a negative delta G, I believe it's around 60 kcal per mole. So we're releasing about 60, not kcal, but kilojoules per mole, which gives it enough energy to transfer the uridine. It's actually UDP over to the glucose, and what this does is now make this molecule a high energy molecule, so that when we add UDPG to the reducing end of glycogen using glycogen synthase, the transfer of the glucose to glycogen and releasing UDP then makes this reaction more spontaneous. Oh, I spelled that wrong. Branching enzyme can then create new branch points as the glycogen molecule is growing. So glycogen is the synthesis or the breakdown of glycogen is regulated through a number of different cofactors. Remember, like I said, glycogen phosphorylase can be activated through phosphorylation. Phosphorylation occurs in the cell initially activated either through glucagon receptors in the liver, or if this is the muscle cell, it's activated through epinephrine. So as glucagon or epinephrine is released into the blood, cell receptors on the outside of the cell will bind that hormone, which will then signal what is called a phosphorylation cascade through, so ATP gets converted into CAMP, which then activates protein kinase. Protein kinase will then activate or phosphorylate phosphorylase kinase, which will then come in and activate glucagon phosphorylase, and then we will get release of glucose 1-phosphate from glucogen. In the opposite, if insulin is released, so we have high blood sugar, so we don't need any glucose from glucagon, insulin will deactivate the adenylate kinase, which stops CMP production, which will then inactivate protein kinase, which then causes dephosphorylation of glucagon phosphorylase. So it becomes deactivated, and we no longer get glucose 1-phosphate cleaved from glycogen. And then we can then activate through dephosphorylation glucogen synthetase, which will then cause glucose to be added to glucogen. So, lots of talking there. But you can read more about this in section 3, a little more in detail, and we'll go over this a little bit in our discussion in class. Okay, so that's the end of this video. The next video we are going to be talking about gluconeogenesis. So this is if we deplete all of our glucogen and our blood sugar, and then we need to make glucose. Gluconeogenesis is how we do that from other precursors.